A biosimilar is a drug that is developed to be similar, as similar as possible to a reference drug, which is a biological drug. The difference to other indications, for example, small molecules where we can generate identical drugs, it's impossible to do this with biological drugs, with antibodies, so they are similar but not identical. There are several biosimilars in clinical development for trastuzumab, and they're at various stages. Some of them are in clinical trials looking at the efficacy, others are in clinical trials just comparing the PK profile and safety. I think there are fundamental differences in the clinical development of biosimilars compared to the reference product. The reference product has to demonstrate a clear benefit in a patient population with clearly defined endpoints that are meaningful to patients. A biosimilar has to be compared to the reference product, not necessarily demonstrating a patient benefit, and has to be compared in terms of safety, in terms of efficacy, and in terms of immunogenicity. I think there are still some challenges around the development of biosimilars. It's the current clinical development plan and the approval process has some challenges. And as I said earlier, it's just comparing safety and efficacy to the reference product. But there are obviously some concerns around how relevant those results are for all indications. Also, there's a limited development program for biosimilars. And there's the possibility for the agencies to approve trials in other indications, even if there are no clinical data available at this stage. So there's an option of extrapolation of clinical data. That has its challenges, and we need to really know whether biosimilars will have the same efficacy compared to the reference drug. It's also then unclear if a biosimilar in indication A shows a comparable efficacy and safety profile, whether this is the case in combination trials. On the other hand, obviously, with the increasing costs of other biologicals and the combinations we are introducing into the clinic, it is a clear consideration that biosimilars may have an advantage in terms of cost efficacy. So we need to work out whether they are safe and as effective as the reference drug. But if that is the case, then there may be advantages in terms of cost efficacy. The challenge at the moment is as always, it's that the devil is in the detail, and we need to look at the small print of those trials. We need to be very careful when we analyze those trials and how we can extrapolate them. So it may be easier to extrapolate trials from early breast cancer into the advanced breast cancer setting, but it's not necessarily possible to extrapolate data from the metastatic breast cancer setting into the early breast cancer setting. And we don't know how the impact of combinations in those in the context of biosimilars will be dealt with. So it is an interesting option. It's something we need to see, and I think there will a lot will happen in the next few years.